Poisoning of the dried and pressed material is also a very important step. So there are a number of agents which may destroy your dried and pressed material, either by the fungi. Some fungus may be destroyed, some insect may be destroyed your collected material. So for that purpose you may use 2% of mercury chloride solution, that is 2% 2% of mercury chloride solution. How to prepare the 2% of mercury chloride solution? The mercury chloride is a granulated form of the chemical, and it is completely dissolvable in absolute alcohol. So you have to dissolve 2 grams. Suppose you are going to prepare the 100 ml of uh, 100 ml of the solution, then you have to add 2 gram of mercury chloride powder in the 100 ml of the absolute alcohol. And similarly you can prepare the stock solution by adding the same quantity of the mercury chloride in the absolute alcohol, and after that you may take out the stock solution and prepare the further solution for the poisoning of your material. This 2% mercury chloride is very dangerous for the insects as well as the eggs or the spores, whatever it is, all or will be destroyed by doing this. Just take out your material, dried and pressed material, and placed in the solution of the 2% mercury chloride, and just take it out, and after some time, because the alcohol is the evaporative, so it will be evaporated, only mercury chloride will remain over there, so that will be sufficient to destroy all the insects, their eggs or the spores, fungal spores, whatever it is. And this by doing this, the your material, dried material will completely be poisoned. Then the next step is the mounting. Mounting is also the important step for the preparation of the herbarium. You have to mount your dried pressed material, which is after the poisoning, you have dried after the poisoning, and you have to mount that poisoned material into the herbarium sheet. And then the question arises, what is the herbarium sheet? How do you define the herbarium sheet? Herbarium sheet is made up of a thick white paper. I repeat, herbarium sheet is made up of a thick white paper with a standard size of 29 and 42 centimeter, having the monogram at the top of the sheet. This is the herbarium sheet. It is not the you can say the official herbarium sheet, but here the monogram must be there at the top of the sheet, and related information at the lower right hand corner. It always be at the lower right hand corner. The institutional monogram at the top of the sheet, related information at the lower right hand corner, and this is the tag number. This bearing the tag number. This tag number will be available at the field notebook, where all the characters of this plant, here you cannot observe what was the color of the leaf, whether the hairs were, it was glabrous or it was hairy, what was the root characters were there. So you cannot observe with the sheets. So all such characters already noted down in the field notebook. That's why the field notebook is important. So we are talking about the herbarium sheet. This is the standard size of the herbarium sheet, that is the 42 and it is 29 centimeter. That is the standard size of the herbarium sheet, and it is of thick white paper. It must be of the color paper cannot use. It must be of thick white paper with a standard size. That is called as the herbarium sheet. On that herbarium sheet, you have to mount a single specimen with each herbarium sheet. Whatever the size of the herbarium, whatever the size of the your plant material. Suppose it is of 2 centimeter. The size of your dried and poison material is 2 centimeter, and the size of your herbarium sheet is 29 to 46, 29 into 42 centimeter. So it means we can place one specimen over here, with one another here, another is here, uh, one more is over here. We can do this, but we can't. We can't do that. Only one specimen must be mounted in a herbarium sheet, whatever the size of the specimen is. If it is more than that size, suppose the size of your herbarium, size of your specimen is the 50 centimeter and it is of 42 centimeter only. How do you arrange? Then you have to arrange with the L shape. Okay. If it is 100 centimeter, what you will do? You have to arrange with the V shape. If it is still unmanageable, then you have to arrange N shape or Z shape, whatever it is. Further, if the specimen, size of your specimen is longer than that, then what you will do? You have to arrange your specimen with W shape or M shape. Okay. These are the mounting techniques. By doing this, you can arrange your specimen with a single sheet. If it is further, if it is longer than the size of the specimen and you are not capable of arranging with these alphabets, then you have to cut down your specimen with the nodes and internodes. Just count down your nodes. Suppose you are preparing the herbarium of a bamboo, so it will not be possible to mount on a herbarium sheet. In that case, you have to count down number of the nodes and internodes and take out at least one internodes and two nodes and kept as such in a glass container. Then if you are preparing the herbarium of the fleshy fruit, then what you will do? You have to kept as such in a glass container with the filled of the liquid preservative, that is of the 4% formalin or FAA. It is a mixture of the solution. What is FAA? It is formalin, acetic acid and absolute alcohol. Suppose you are preparing 100 ml of FAA, then you have to 90 ml of absolute alcohol, 5 ml of acetic acid and then 5 ml of formalin. By doing this, you will get 100 ml of FAA. and all the fleshy fruits, the dry fleshy fruits, may be kept in a liquid preservative, and just write down the tag number, whatever it is, in that bottle, and all the dried material. For example, it is of palm inflorescence, or it is of cycas cone, or it is of pine cone, whatever it is, it may be dried and kept as such in a glass container after the poisoning, whatever it is. Then after doing this process, we can mount the specimen on a herbarium sheet. Then the next step is to how to this is the labeling of the herbarium. How would you label? You have to identify the material. What is the botanical name? We have identified which family it belongs. What was the local name? What was the locality? It was noted down over there. I said the lower right hand corner, the information must be available over there. So such informations we always be available over there. These are the informations. These are the information related to that plant is over there. Then the filing of the herbarium sheet. Suppose this is your herbarium sheet. This is one of your herbarium sheet. How would you file your herbarium sheet? Suppose, sorry, these are your herbarium sheets which you have prepared after pressing, mounting, poisoning. It is complete herbarium sheet. All the steps you have followed. Then you have to file this. Suppose the members of the convolvulaceae you have collected. In convolvulaceae you have collected about 5 or 7 species of the same genus. Suppose it is Palmata, Ipomia, Pestigridis, then Ipomia, Comaclit, then Ipomia aquatica. So suppose these 5-7 species of the same genus you have collected. So all such species of the same genus may be sorted out and be placed in a folder like that. This is one of the suppose it is Ipomia pestigridis, this is Ipomia comaclit, and this is Ipomia aquatica. Whatever the species of the Ipomia is must be placed in a folder and you have to name this folder as genus folder. You have to write over here the Ipomia. Okay, so this would be called as genus folder. Then in convolvulaceae, suppose you have collected about 10 genera. So all these genus folder must be placed in a separate folder that will be called as family folder. You have to write over here the family folder. Which family you have to write over here? Convolvulaceae you have to write over here. If any other worker wanted to study the material of the convolvulaceae, he will take out the family folder of the convolvulaceae from your herbarium almirah and he will go through the different genera whatever you have placed in your family folder, and again he got the particular genre for which he was interested to study. Suppose he is studying in the Ipomia species, so just he take out the genus folder and he will tally his material with all the Ipomia species are placed over here. So this is the system of the filing system of the herbarium sheets. And these family folders be always placed in a, this is the family folder. This is the Nephilium, that is the genus. This called as the genus folder, and all the Nephilium species are placed inside this genus folder, and then it will go through the another folder that is called as the family folder. This is all the family folders are there, and it is placed in a pigeonhole almirah. And that almirah, this is the pigeonhole almirah. So these almirahs are arranged according to the Bentham and Hooker system. In the Bentham and Hooker system, the starting family is the Ranunculaceae. So in the first column here, in the first column, you have to put all the family members of the Ranunculaceae. Okay, and in the last, there are a number of the pigeonhole almirah is available over there. It is depending upon the monetary condition of the institute. If it is single almirah, pigeonhole almirah provided to you, then you have to arrange your family folders in only limited columns. Instead of the Ranunculaceae, you may place 2 or 3 family folders in a single column, or in a separate column only if the financial condition is good in your institute where you are working. So there may be some more almirahs may be provided to you, so you may use one column for the one family depending upon the situation. It is up to you what the situation you are facing over there. But my point is, all the family folders must be arranged according to some approved system of classification. So which system is approved? As early in the starting I have told you that only in the Bentham and Hooker system is approved in India. So the Ranunculaceae must be placed in the starting, as well as the ending family, which is the, that is the Glumaceae, or the order is the Glumaceae and the family is the Poaceae. Poaceae and Cyperaceae comes under this order. So all the members of the family folder, the Poaceae family may be placed over here. So this is the complete information about the preparation of the herbarium. And then the herbarium care is also needed. The accession number, if your herbarium is recognized by the institution as well as the government, then you may give the accession number. Accession number, each specimen will have a particular number, so that be cited out if anybody is doing research and contacted your herbarium, then he will have to mention the accession number. And then the herbarium care. Though you have poisoned your plant material with the 2% mercurochloride, but still there is chances to have some insect effect. So that care must be there. For that purpose, we can use the DDT, we can use the DDT, we can use the carbon disulfide gas, we can use the naphthalene balls, we can use the paradichlorobenzene. We have to place these paradichlorobenzene or DDT or whatever it is in each column. Even the naphthalene balls, you can place some naphthalene balls in the each column of the pigeonhole almirah or paradichlorobenzene, or you can fumigate with the carbon disulfide gas, whatever it is. You can use these chemicals for the caring of the herbarium. If your institution is not able to provide these chemicals for the caring of the herbarium, even you can use the margosa leaf, the Zadiracta indica leaf, or you can see, you can known as the neem leaf. Neem leaf will also serve the purpose up to some extent. So by doing that, you can care your herbarium. This is the liquid preservative is used for the mostly for the fleshy foods collection. So thank you so much. The next topic we will discuss in the next session.